Hello everyone, my name is Sonia Sladia. I'm a PhD candidate at Clemson University. Today I'm here to present my master's research, which is about evaluation of an interspecific cross between cultivated chickpea and a wild species that is Cicer pinnatifidum. So I'll be introducing a little bit about chickpea. So chickpea is a self-pollinated legume crop which was originated in Fertile Crescent. This crop was domesticated 7,000 years ago, and presently it is being cultivated in more than 50 countries, and India is leading in its production. So we all know it is a legume crop which fixes atmospheric nitrogen through the rhizobium bacteria residing in its root nodules. So these bacteria maintain a symbiotic relationship with the plant and fulfill the nitrogen needs for the plants. And lastly, chickpeas are nutritionally very dense. They're having a good amount of plant protein. So, because of that, they can be used as a raw material for different plant-based products. They can be consumed in different forms. They can be boiled, they can be roasted, and they can be processed to make hummus, protein powder, and other products. In chickpeas, there are annual species which are classified into three groups depending upon the ease of crossability. So, in the first group, there are three species, including the cultivated one, and these species are easily crossable with each other. And then we go to the group two. Again, there are three species. These species are also crossable with the group one, but they may sometimes need some hormonal treatment or maybe embryo rescue, but mostly result in the fertile hybrids. But when we come to the group three, these species are difficult to cross with the group one, and to recover the hybrid successfully, you definitely mandatory, maybe need to use some specific techniques, as I mentioned before, like embryo rescue, embryo culture, and growth hormones as well, and also sometimes the bridging species to make the hybrid successful. Now, coming to why did we go for the interspecific hybridization? So the two main points are, the first is to broaden the genetic base of chickpea. So since it is a self-pollinated crop, so naturally there is not much variation for the different traits. So to expand the variation, we need to go to the wild species. Now, the another reason is to explore for the new alleles. So when we see that the present variation we have is not enough, we go to the wild species and explore for the different traits like phenological traits, productivity traits, and disease resistance. And particularly wild species are good source for disease resistant genes. So now I will be telling that how this all work was started. So in 2010, 2011, the chickpea breeding team at Punjab Agriculture University in India, they evaluated several wild species and accessions from them for the two diseases, that's Ascochyta blight and botrytis gray mold. And from all those different species, there was one accession belonged to the Cystopenatifidum, which was showing a good level of resistance for both the diseases. And this was chosen as a male parent to develop a population. So, with that, for the population development, we had a female parent, which is a bushy mutant. And this bushy mutant was spontaneously originated from one of the crosses that breeding team was already having. So, this female parent was crossed with the male parent, and we recovered an F1. So this F1 was confirmed before it was proceeded for the advancement. So for confirmation of this F1, several traits were recorded in it and compared with the parents. So in this table, particularly you can see the different traits in the first column, then the traits recorded in the female parent, in the male parent, and in the far right column you can see the traits recorded in the F1 hybrid. So for the traits which are highlighted in the purple here, they were corresponding to the traits which were observed in the male parent. And the other traits which are highlighted in the orange color, they were corresponding to the female parent. So this is a quite a good observation to confirm that it is a true hybrid. Then after confirming the hybrid is true, the F1 was advanced to the F7 generation by following the pedigree method. And at F7 generation, some of the superior derivatives are selected for further study. So for this particular experiment, we chose 158 interspecific derivatives and they were tested along with the two parents for the 2 years in 2018 and 2019. So in here, it is just an illustration that how the experiment would have been looking like. So the green plants here represent the interspecific derivatives, and the red one is representing, say, a female parent, and the black one is representing, say, a male parent. So for experimental design, so we had a field experiment which was laid out in alpha lattice design with two replications in it, and this was scored for phenological traits and productivity traits. So we scored it for two phenological traits and seven productivity traits. And the same material was also used for disease resistance screening. For that, we had a field experiment which was laid out in RCBD, randomized complete block design in two replications for ascochyta blight screening. Then for botrytis gray mold screening, we have a greenhouse experiment, which was laid out in completely randomized design, and that too in two replications. For the disease screening of ascochyta blight, we had a disease rating scale, which was described by Reddy and Singh. So according to this scale, the lowest number indicates the good genotype, which has a good level of resistance, and the highest number indicates the poor genotype, which was susceptible. So in this scale, you can clearly see that the genotype with number 1 is showing no symptoms of the disease, and as you progress from 1 to 9, the symptoms just getting worse, and this plant is look like completely devastated. Now, similarly for the botrytis gray mold, we had a similar scale, but it was devised by Singh and co-workers. And similarly, in this scale also, the lower number indicates the good genotype, good level of resistance, and the highest number indicates the susceptible derivatives. So here you can see a range we observed for botrytis gray mold as well. So now coming to the how the different derivatives were looking like in the fields. So this is a field view or the experimental view where we tested the different derivatives. In this particular picture, you can see the different growth habits of different derivatives. So there were two parental types, the bushy one and the prostrate one. Other than that, there were other four intermediate types of growth habits observed in different derivatives. And the most of the derivatives were belonging to the semi-spreading class, and the least number were present in the prostrate class, which was similar to the male parent. Similarly, we also recorded variability for other morphological traits, such as flower color. So here you can see the lighter flower color in the female parent and the darker color in the male parent. And this is a spectrum of variation you see in the derivatives. And when we see the leaf size and leaf serration, so leaf size was more in female parent compared to the male parent, but serrations were not that deep. They were very narrow serrations. So in the male parent, the serrations are very, very deep. But in the derivatives, we see a kind of different kind of variation and intermediate types for the different leaf size and leaf serrations. Now we come to the leaf color. So in the female parent, we have lighter color compared to the male parent. And this is the variation we found in the derivatives. And when we come to the pod size and pod shape, so in the male parent, you can see the pod is smaller, it's narrow, it's elongated. But in the female parent, the pod is more rounder and it is a medium in size. And this is the size comparison of different types of pods we have found in the derivatives. So all these evidences were quite good enough to confirm that this is a true cross and true material developed. Then we uh, then we also recorded the different phenological and productivity traits. So we did analysis of variance. In the pooled analysis of variance, we found all the different genotypes were significantly different for all the traits. We also had mean values for all the different derivatives, we compared them with the female and male parents, and then we also had a range for different traits. So in this particular table, the orange colored, these light orange colored highlighted cells, these are indicating the higher mean values than both the parents, and this blue one is indicating the lower mean value than both the parents. So this is kind of indicating a transgressive segregation we have observed. And particularly, I would like to bring to notice that these traits are the productivity traits. So you can see here the productivity traits are having higher values than both the parents. So which is a positive thing. Now coming to the disease reaction in a disease reaction or disease screening we observed for Escocata blight. So here you can clearly see that both the parents are far apart for this disease reaction. So Cicer pinnatifidum, which is a wild parent, it is quite good, it's quite resistant, but the female parent, it's so much susceptible, it's all devastated. And after screening of all the derivatives, we found this particular derivative which was resistant, and this derivative which was kind of showing moderately resistance to the Escocata blight. Then similarly for botrytis gray mold, we found out Cicerpinnatifidum is resistant and Bushy mutant is susceptible. And when we had a screening in the greenhouse, so we found this is a range of different types of reactions to the botrytis gray mold by the different derivatives. So here is the susceptible plants and here is the resistant, but I would not say these were resistant, these were moderately resistant actually. Then coming to the another part of this experiment, which was used to confirm the introgression using molecular data. So in here, we extracted the DNA from all the derivatives and parents by using the CTAB method. And that DNA was sent out to get the genotypic data by using genotyping by sequencing technique, and then was processed to get the polymorphic SNPs and to perform the graphical genotyping. So this is a distribution of different SNP markers which were polymorphic on the different chromosomes. So there were total 1400 polymorphic markers, and most of the markers were present in the chromosome number 4, probably because it's the largest chromosome recorded so far, and the lesser number of markers were present in chromosome number 8, and because it's a smaller in size, so it could be the reason. Now coming to how much percent introgression was happened from wild parent to all the chromosomes. So in this particular figure, the different pie graphs, they're having red color, which is indicating the alleles from Bushy mutant. Blue color is indicating the alleles from Cicer pinnatifidum. That's a wild parent. And the green color is indicating the heterozygous loci for both the parents. So in here, we observe that on chromosome number 7, most of the alleles were from wild parent, and it could be due to the more recombination at this particular chromosome compared to others, and the least introgression was found on the chromosome number 8, again possibly because it is smaller in size. Now coming to the point that how this distribution was in the different derivatives. So all the derivatives were categorized into different classes, and most of the derivatives were having uh, introgression from 34% to 38% from wild parent. And highest introgression was recorded in this particular derivative, and the lowest was found in this particular derivative. Then at last, coming to the what are the salient findings. So we observed a big whole range of variation in morphological traits. That was a good information to confirm that this is a 2-cross. And again, to confirm it at molecular level, we performed graphical genotyping, which was again successful. And then we have observed high mean values for most of the productivity traits, and also it indicates a transgressive segregation. And for yield traits, we found these 3 particular derivatives which were promising. Then for disease screening, we found only one derivative resistant to Ascocata blight, And these four derivatives were having moderately resistant reaction and possibly the lowest score we had. Now, for botrytis gray mold, we weren't able to get any uh, resistant derivative, but we found these four derivatives to be moderately resistant. So these could be used for the future breeding programs, which they are using, possibly. Now, at last, I would like to acknowledge Punjab Agricultural University for providing the funding and support to carry out this research. And I also would like to acknowledge my mentor, Dr. Sarvjit Singh, for his able guidance throughout this work and my journey. And at last, I would like to acknowledge Dr. Dharmender Bhatia for helping me with the analysis. I would also like to thank the meeting organizers for inviting me as a guest speaker. I will be happy to answer all the questions. Thank you so much.